Hi group, and welcome to Unit 6, where we're going to talk about the family Micrococciaceae. And anytime it ends in ACA, we're referring to a family of organisms. Micrococciaceae includes organisms Planococcus, Micrococcus, Stomatococcus, and Staphylococcus. Okay, there are about 7,000 known Micrococcus and Staphylococcus species, and there's probably over 100,000 yet to be identified. So let's go ahead and take a look at the Micrococcus and the Staphylococcus. All right, so there's two organisms I want to start with. That would be Planococcus and Micrococcus. We're not going to talk about Planococcus too much, just a little bit right now, and then that'll be the last you hear of Planococcus. But Planococcus is unlikely source of human infections, and there are 9 species, and that's about all you need to know about Planococcus. The one that I want to focus on next is Micrococcus species. There's actually 2 species, Micrococcus luteus and Micrococcus roseus, and we worked with both of those organisms in Lab 1. Okay, there has been several taxonomic reorganizations to this genus over the last couple years. Like I said, there are 2 species, Micrococcus luteus, which is yellow or gray, or Micrococcus roseus, which is salmon or pink. Okay. All right, Micrococcus luteus is the most commonly isolated micrococcus in clinical specimens, and that's what you're going to see more than anything else out of the micrococcus in lab. All right, I want to talk about Stomatococcus mucilaginosus, and that has got a new name for this genus. The new name is called Rothia, R-O-T-H-I-A, Rothia genus. It's the only member of the genus Rothia, and it is part of the normal respiratory flora of humans. And that is actually the new name now. It's Rothia. Rothia is a gram-positive organism. It's easily noted in upper respiratory cultures because it can be pulled off the agar in one piece with a stick or a loop. And you can kind of see that in the image on the right. When we take a loop and we sort of tease that colony, it kind of comes up like an egg white. Now, Staphylococcus is an important genus that I want to talk about, and we're going to be seeing a lot of Staphylococcus in student lab. It is a frequently isolated organism in clinical specimens, and there are several species. Some, of course, are considered to be normal flora, and some are pathogens, and sometimes they can be both. It just sort of depends on the host that they are in or on. Okay, it is the second most frequently isolated microorganism in clinical specimens, and it is widespread in nature. And sometimes it can actually contaminate our plates when we're trying to grow something else because it is on our skin and on our hands. Okay, certain toxins can be responsible for food poisoning, such as enterotoxin A and D. And then also certain toxins can be responsible for toxic shock syndrome, like TSST1, which is the toxic shock syndrome toxin 1. But some of these Staphylococcus do make toxins. Okay, out of all the Staphylococcus, Staphylococcus aureus is probably the most important clinically. So going back a little bit on the slide, we're going to talk about the Micrococciaceae, and we're going to talk about all of the characteristics that are common to these organisms. Okay. One such organism, of course, is Staphylococcus. Staphyl is Greek for bunches of grapes. So when you see the word Staphylococcus, it basically means that it gram-stains in clusters. Okay, so out of all the Micrococciaceae, some of the characteristics that are common is that they are gram-positive cocci, and sometimes they can be in pairs. Other times they can be in tetrads, like Micrococcus is often in tetrads. Sometimes they can be in clusters, clumps, or packets, and usually that's Staphylococcus. They are aerobic or facultative. They can be facultative anaerobes, so in other words, they prefer no oxygen, but because of the cost and the expense that it would cost us to prepare them anaerobically, we typically grow these aerobically, but they are facultative anaerobes. They're not fastidious, they don't require any special media or anything like that, and they're non-modal. They're catalase positive, and I'm going to talk about what catalase is in the future. It's basically an enzyme that's present in Staphylococcus and Micrococcus, and they do reduce nitrates to nitrites, and we're going to talk more about that in the future as well. In a broth, Micrococciaceae will appear as streamers, and there's some nice pictures in your book about that as well. So in this slide, you can see a picture of staphylococcus that are gram-positive cocci in clusters. So gram-positive means they're purple, they have a thick peptidoglycan wall, and they are forming clusters. Staphyl, of course, means cluster of grapes in Greek. And so you can see that they are cocci, which means that they are round or circular objects. So they're gram-positive cocci in clusters. And then you can see the streamers of staphylococcus growing in a broth. You can see how it grows these lines in the broth. That is very typical of staphylococcus. Now, organisms that make us sick will produce certain virulence factors, and a virulence factor is something that makes it more pathogenic. And in terms of micrococciaceae, you're going to see that some of these organisms produce enzymes and toxins. So some enzymes that are produced would be lipase. Lipase, of course, is like the oil on our skin that promotes the growth of staphylococcus. Hyaluronidase is another enzyme that dissolves connective tissue. Staphylokinase is another enzyme that dissolves clots. Coagulase takes fibrin and uses it to surround organisms so that they cannot be phagocytized. And beta-lactamase organisms are positive for resistance when it comes to the beta-lactam drugs. So if you have an organism that's beta-lactamase positive, it's actually resistant to penicillin, ampicillin, and the first generation cephalosporins. That brings us to the toxins. There are several toxins produced by micrococciaceae that make it more pathogenic for the host. We've got leukocidin, which destroys white cells. Enterotoxins, which are toxins that cause things like food poisoning. Okay, and then we've got epidermolytic toxins, which are toxins that are on the skin, such as exfoliative toxin, which actually destroys skin cells. We have the cytolytic toxins, which destroy certain cellular items, for example, platelets. And then we've got toxic proteins like protein A. Protein A actually binds to immunoglobulins, so it can't cooperate with phagocytes. So that's kind of a list of the enzymes and toxins that make staphylococcus and micrococcus especially pathogenic. And then here, I guess I didn't realize I had this slide, it just kind of summarizes everything that I said in the last slide. So just take some time to look at that. So in the laboratory, we are always tasked with proving what the name of our microorganism is. And so there's a bunch of tests that we can do to determine the genus and species of our organism in the family Micrococciaceae. So the tests that we can do are catalase tests, nitrate reduction, glucose fermentation tests, lysostaphin tests, microdase tests. We can test for resistance or sensitivity to bacitracin, furazolidone, and novobiacin. We can determine if they grow on mannitol salt agar. We can determine if they're coagulase positive or negative, if they're beta-lactamase positive or negative, and if they are resistant to oxacillin. Now the new name, the old name for oxacillin was methicillin, but it's the same thing as oxacillin. So we're going to go ahead and talk about all of these tests, and these are the tests that we can use to identify our Micrococciaceae down to the genus and species level. So I first want to talk about the catalase test, and the catalase test uses a reagent known as 3% H2O2, which is essentially hydrogen peroxide. Okay, catalase test is often used to differentiate between aerobic gram-positive cocci like Staphylococcus, which is catalase positive, and Streptococcus, which are catalase negative. Okay, it determines the presence of the catalase enzyme. If you add 3% H2O2 with an organism and it starts to bubble, it means that your organism has the catalase enzyme. Okay? The principle is organisms that are catalase positive will convert 3% H2O2 to water and oxygen gas, which is demonstrated in bubbling. Okay? Now, whenever you do this test, we want to be cautious not to get chunks of our blood agar in it, because our blood agar contains peroxidase, and that's going to give you a false positive. So you want to make sure that when you do this test, you get just the organism and not any chunk of the blood agar. Now, Staphylococcus and Micrococcus can reduce nitrates to nitrites. Nitrates are actually in the environment, and we can determine the ability of an organism to reduce nitrates, that's NO3, to nitrites, which is NO2, or reduce nitrates to nitrogen gas, which is N2. Staph and micrococcus will reduce nitrates to nitrites. Okay, the reagents used in this test are alpha-naphthalene and sulfonylate acid, and then we use a third reagent known as zinc. So if you take a look at that picture on the bottom right, you can see that the two tubes on the right are negative for nitrate reduction, and the two tubes on the left are positive for nitrate reduction. So essentially what I've done is I've added equal parts of alpha-naphthalene and equal parts of sulfonylate acid to my tube that's growing my staphylococcus or micrococcus. If it turns red, it means it's reduced nitrates to nitrites. Now, if it has not reduced nitrates to nitrites, we can add a little bit of zinc dust. If the zinc dust turns it pink, then it is negative. It means nitrates have not been reduced. If the zinc dust stays clear, like if it does not turn pink, it means that our bug has reduced nitrates to nitrogen gas, and that's still a positive test. So this is kind of a complicated test. We will do this test in the laboratory several times, so don't worry if you didn't get all that down right away. Now, some of our staphs and micrococcus will ferment glucose, okay? So we can use a media known as OF glucose or uniglucose. The tubes on the right is a picture of uniglucose test, but OF glucose test looks very similar, only it doesn't have a pinching of the glass in the middle. Okay, but what we can do is we can add our organism to the tube using a stick or a loop, and if the organism turns yellow at the bottom, it means that it's a fermenter, and our staphylococcus are glucose fermenters. Micrococcus is not a glucose fermenter. Micrococcus is actually negative for glucose fermentation. So when staphylococcus ferments glucose, what's going to happen is that there are acids that are being made because of the fermentation of that sugar. So the pH indicator, which is bromthiamyl blue, will actually turn yellow. Okay? Micrococcus does not ferment glucose, and so the media remains alkaline, or that's demonstrated by a bluish color, or it's going to remain inert and it will stay green. Now, some staphylococcus produce small amounts of acids, which makes the media turn yellow very slowly. So some of these weaker results would be from organisms like Staphylococcus saprophyticus. Okay, so they may require a longer incubation period to get that glucose tube to turn yellow. Remember, it's turning at the bottom. Fermentation is a process that does not require oxygen, so you're going to see it demonstrated in the base of the tube, and oxidation is something that occurs in the presence of oxygen, and so you would see the color, the yellow color turn in the top of the tube. So here we're looking for glucose fermentation, which is yellow coloring at the bottom of the tube. The microdase test is considered a modified oxidase test, and the reagent that we use for the microdase test is called 6% tetramethyl p-phenylenediamine. Now, do you have to spell that? No. Do you have to recognize it as being part of the microdase test? Yes, you do. Okay, so what we do in this test is we take a little disc, and this disc is impregnated with 6% tetramethyl p-phenylenediamine. And we take our organism and we actually rub it on the microdase disc. And we don't moisten the disc or anything like that, but then we set a timer for 30 seconds. At the end of 30 seconds, if the disc turns purple, then it's positive. Micrococcus is going to be positive for the microdase test, and Staphylococcus are microdase negative. So here we use the microdase test to determine if we have a Staphylococcus versus a Micrococcus genus. The lysostaphin test is not one commonly done anymore, but I do want to talk about it, although we won't be doing this test in lab. Okay? The lysostaphin test is made from toxins extracted from Staphylococcus staphylolyticus. And basically what it does is it differentiates between Staphylococcus and Micrococcus. So what we do is we make a heavy suspension or a heavy turbid suspension of our organism in the lysostaphin reagent. We incubate for several hours. At the end of our incubation, if it stays cloudy, it means that our bug was not lysed. So that is a negative test. So cloudy means negative, and that would be a confirmatory test for Micrococcus luteus. If the suspension is clear, like it is in the bottom left image there, you can see that it is a positive test, and that would mean it's Staphylococcus. So basically what I'm saying on this slide is if you take any Staphylococcus species and you combine it with Staphylococcus staphylolyticus, if it lyses, it's going to be a positive test. If it does not lyse, it's going to be a negative test. Now, this is definitely a test you're going to want to know. This is the coagulase test, and it's the best way to identify the most prominent Staphylococcus pathogen, which is Staphylococcus aureus. Okay, there are two methods available, a slide method and a tube method. I prefer the tube method, but some of you might prefer the slide method, and we'll do both. Okay, but basically what we have is the coagulase enzyme present in Staphylococcus aureus that has reacting factor and fibrinogen, and it forms a fibrin clot. Okay, clot formation is considered a positive test. So if you add your organism to the coagulase reagent and it clots, that is a positive test. The only organism that's going to be positive for coagulase is Staphylococcus aureus. Okay, and then we have the bound coagulase test, where we have latex agglutination coagulase procedures using latex beads coated with fibrinogen to detect clumping factor. If you have bound coagulase or clumping factor that is attached to bacterial walls, it's going to be positive for the latex agglutination bound coagulase test. Okay, so a fibrin clot is going to be seen with rabid plasma. That is the reagent in the coagulase test for Staphylococcus aureus. 99% of Staph aureus organisms are coag positive, and other species are coag negative. So for all the other species of Staphylococcus, we call those coag negative staphs. And then, of course, Staphylococcus aureus is our coag positive staph. And then, of course, you need to know the reagent for the tube coag test, and the reagent is rabid plasma. So rabid plasma is the reagent for the coagulase test, as it contains coagulase reacting factor. So if you have coagulase reacting factor and coagulase, it forms a visible fibrin clot. We can also use some substances on a disc to determine if we have a Staphylococcus versus a micrococcus genus. Okay, so this next slide I want to talk about is the furazolidone disc. So basically we have a plate of our micrococcus and a plate of our Staphylococcus, and we can actually drop this disc on our plate, then incubate it. At the end of our incubation, if the zone size measures 15 millimeters or greater, then it's got to be a Staphylococcus because they're sensitive to furazolidone. If the size of the zone is 6 to 9 millimeters, then it is considered resistant, and that has to be a micrococcus. So by using the furazolidone disc, we can determine if we have a Staphylococcus genus, which is going to be sensitive, or we have a micrococcus genus, which is going to be resistant to furazolidone. And we're going to be doing this in lab. The bacitracin disc has a couple of applications. It's also known as the A disc because we also use it to identify Streptococcus group A. But we're not talking about that today. Instead, we're talking about the A disc in terms of using it to identify Staphylococcus genus versus micrococcus genus. So Staphylococcus are resistant and they grow right up to the disc, so bacitracin is a disc, and micrococcus is sensitive. So the zone size is going to be greater than 10 millimeters. Okay, so again, we have a plate of our organisms. We put the disc on our plate and incubate it. And if you have a zone size that is very small, it's a Staphylococcus genus. If you have a zone size, a clear zone size that's greater than 10 millimeters, that is going to be sensitive and it's going to be a micrococcus. Novobiocin is a disc that we only use to determine if we have a Staphylococcus saprophyticus versus a Staphylococcus epidermidis. Okay, so Staphylococcus saprophyticus is actually resistant to novobiocin. Okay, so again, we're going to take a plate of our organism and we're going to drop that novobiocin disc. If the zone size is 6 to 12 millimeters, then we're going to say it is resistant and it's going to be a Staphylococcus saprophyticus. If the zone size is greater than 12 millimeters, then it's sensitive and it's Staphylococcus epidermidis. So it is important to speciate Staphylococcus because Staphylococcus saprophyticus is one of the leading causes of urinary tract infections among females of childbearing age. Now, like I said, all of the Staphylococcus that are not Staph aureus are considered coag negative staphs. So Staphylococcus saprophyticus is one of several coag negative staphs. Now, mannitol fermentation is something I want to discuss because our staphylococcus can ferment mannitol, or some of them can, and they will grow in the presence of 7.5% sodium chloride. So mannitol salt agar, also known as MSA, is both selective and differential. It is selective for staphylococcus, and it's differential because we can determine if our organism ferments mannitol or it doesn't. Okay, so staphylococcus can tolerate 7.5% sodium chloride, so all staphylococcus will grow on mannitol salt agar. It's differential because only some of the staphylococcus are mannitol fermenters. So if our organism turns yellow, it means that it is a mannitol fermenter, and one strong mannitol fermenter is Staphylococcus aureus. If the organism stays pink, then it tolerates salt, but it does not ferment mannitol. So there are some examples of organisms that do not ferment mannitol, and those would be other Staphylococcus species. Now, micrococcus will not grow on mannitol salt agar at all because it cannot tolerate 7.5% sodium chloride. The beta-lactamase test is a test to quickly determine if our organism is sensitive or resistant to ampicillin, penicillin, and the first-generation cephalosporins. It's a disc, and what we do is we take our organism and we smear it on that disc, and then we wait anywhere between 15 and 30 minutes. If the disc changes color depending on the manufacturer of the disc. If it changes color, it means that your organism is resistant to the beta-lactam drugs. And those beta-lactam drugs, again, are penicillin, ampicillin, and first-generation cephalosporins. So usually we use the beta-lactamase test for Staphylococcus aureus and sometimes other Staphylococcus genus as well, but we can actually do the beta-lactamase test on a number of different organisms, including some of our Neisseриas and our Enterococcus faecalis. Now, anytime you isolate a Staphylococcus aureus in the lab, we want to make sure that it's not an MRSA. And I'm sure you've all heard of MRSA before. It's a very resistant strain of Staphylococcus aureus. It means that it is methicillin resistant. And methicillin is the old name for oxacillin. Okay, so let's talk a little bit about the timeline of how we've seen methicillin resistance. So in 1944, we saw the first penicillin-resistant organisms. So they invented ampicillin, methicillin, and drugs like that. In 1951, methicillin is invented, and in 1952, we started seeing methicillin resistance already. By 1975, 2% of all Staph aureus were MRSA, and by 1996, 35% of Staph aureus are MRSA, and I'm sure that's got to be closer to 50% at this day and age. Okay? Staph aureus infections can sometimes be easily treated with penicillin drugs, the beta-lactam drugs, but other times we have to isolate Staph aureus because they are very resistant and they're not going to respond to the beta-lactam drugs. Okay, there are a bunch of tests that exist to determine if we have an MRSA. One thing you can do is you can take Staph aureus and put it on a Mueller-Hinton plate that's got about 4 micrograms per mil of oxacillin. If your organism grows, then it means it's resistant to oxacillin, thus it's an MRSA. Micrococcus luteus is normally insignificant, and you see it quite a bit in places like the upper respiratory tract or on your skin. Okay, they look pretty dull. They don't look damp or wet at all, and they are normal skin flora. Most strains are yellow or gray, and they are slow growers. They're catalase positive. All of your staphs and micrococcus are catalase positive. They're glucose fermentation negative, lysostaphin negative, microdase positive, and they will not tolerate high amounts of sodium chloride in the mannitol salt agar. And then they are sensitive to the A disc and resistant to the furazolidone disc. So Micrococcus luteus are normally insignificant. We generally don't have to identify them down to the species level. We do in student lab, but in the clinical world, we generally do not because they are usually insignificant. The one that we have in student lab, the Micrococcus luteus we have in student lab, is bright yellow. Coag-negative Staphylococcus refers to any Staphylococcus that is not Staphylococcus aureus. So the two that we work with in laboratory are Staphylococcus saprophyticus and Staphylococcus epidermidis. Both of these are considered to be coag-negative staphs. Okay? They can be the cause of miscellaneous infections and nosocomial infections, although they are common plate contaminants and they are skin flora. Okay, but they can cause other things like urinary tract infections and things like that. They can cause heart valve infections and things like that as well. They may contribute to wound infections, bacteremia and sepsis, osteomyelitis, which is a bone infection, and catheter infections. It may also be associated with biofilms, and biofilm is a resistant community of organisms that live together and act as a whole. Staphylococcus saprophyticus is significant in the fact that it can cause urinary tract infections in women of childbearing age or sexually active females. These urinary tract infections can lead to inflammation or infection of the bladder, which is known as cystitis. Okay? If we want to speciate our Staphylococcus, we can drop a novobiocin disc. If it is sensitive, it's Staphylococcus epidermidis. If it is resistant, it is Staphylococcus saprophyticus. And determining the actual species of the Staphylococcus can be important when we're looking at the clinical picture. Okay. Most of these Staphylococcus saprophyticus are gray to white, medium-sized opaque colonies, and it is one of our coag-negative staphs. It's usually gamma hemolytic. Gamma hemolytic means that it does not lyse the red cells in the blood agar plates. So it's considered gamma hemolytic or non-hemolytic. Staphylococcus epidermidis is another coag-negative staph, and it can cause things like nosocomial infections, which is a hospital-acquired infection, or endocarditis. It can be surprisingly resistant to antibiotics, and it is often a contaminant on your plates because it's skin flora. It is sensitive to novobiocin. Remember, we use novobiocin to determine if we have a staph saprophyticus versus a staph epidermidis, and staph epidermidis is sensitive to novobiocin. They are sometimes bright white, opaque, medium-sized colonies. Once in a while they'll look beta hemolytic, but for this class we're going to say they're gamma hemolytic most of the time, gamma hemolytic. Stomatococcus, the new word from Stomatococcus is Rothia, and it's part of the normal upper respiratory flora. Usually it's pretty recognizable and we don't go on to identify it to the species level. Instead, what we do is we take a loop or a stick and we tease it off the agar, and if it pulls all the way off like an egg white, we just call it a Rothia or Stomatococcus species. So I think you'll find this slide very helpful for you in laboratory, okay? How do we determine if we have a Staphylococcus versus a micrococcus, okay? Micrococcus are microdase positive, lysostaphin negative, glucose fermentation negative, and won't tolerate 7.5% sodium chloride in a mannitol salt agar. Staphylococcus, on the other hand, is microdase negative, lysostaphin positive, and glucose fermentation positive. So those tests that I just mentioned are how we determine the genus if we have a micrococcus or a staphylococcus. If we want to further name our organism down to the species level, we could do a coagulase or mannitol fermentation. And Staphylococcus aureus is coag positive and mannitol fermentation positive. It grows very easily in the presence of 7.5% sodium chloride, and it's actually going to be yellow because it ferments mannitol on that plate. Staph saprophyticus is resistant to novobiocin, and Staphylococcus epidermidis is sensitive to novobiocin. All staphs and micrococcus are catalase positive. Remember, we use the catalase to determine if we have a Staphylococcus versus a Streptococcus. Streptococcus are catalase negative. Anyway, I would recommend thumbing through your book and finding the flow chart that talks about how to differentiate between Staphylococcus, Streptococcus, and micrococcus, and I would probably make your own flow chart that you can bring to lab. If you want to use the book flow chart, that's fine too, but that's the best way to go about learning how to identify the Staphylococcus and micrococcus. So that's it. That's Unit 6. If you have any questions, just give me a shout.